It's really my pleasure and honor to introduce this year's Pritzker Award winner, Dr. Linda Griffith. Linda is the School of Engineering Professor of Teaching Innovation in the Department of Biological Engineering at MIT, and she's widely recognized for her groundbreaking work in biological engineering, blending cutting-edge research with a commitment to advancing education and innovation. She's a member of the National Academy of Engineering and was elected into the National Academy of Medicine for her pioneering work in tissue engineering, biomaterials, and systems biology, including developing the first liver chip technology. She's also a founding member of MIT's Biological Engineering Department. So it's my pleasure to welcome Linda to the stage.
It's really great to be here with all my BMES family and share with you a little bit about work that led up to this idea that we may someday move over the mice and replace them with living patient avatars. And to start off and frame the themes for the main part of the talk, I want to go way back to when I started my faculty career, right after I finished a postdoc with Bob Langer. And it was an era in tissue engineering of phenomenology. And this is a great example of that. This was one of the first papers from my lab, published in Materials Research Society Proceedings, in which the innovation was making a degradable polymer in the shape of an ear. So you could put cartilage cells on it, implant it in a mouse, and grow a piece of cartilage in the shape of a human ear. Now, this inspired a lot of people to go into the field, but it wasn't really a template for how you could do that for more complex organs, things like liver, bone, and so on, because those kinds of cells needed a lot more instructions than just some empty space to guide them into forming tissue in vivo. And so this sparked the era in the whole field, but certainly in my own lab as I was starting it, to think about design principles for polymeric biomaterials. And I cite this paper here by Gargi Maheshwari. She was the first graduate student that was joint between me and Doug Laufenberger after he moved to MIT in 1995. And you can see she's giving a plenary talk tomorrow afternoon for her award. But what was really exciting at that time is molecular biologists were understanding the rules of how cell surface receptors bound to specific regions of extracellular matrix. And Doug Laufenberger was figuring out how do those actually work biophysically to govern, for example, the speed of motility. And this gave us design principles to go in and say, okay, we can't use fibronectin in vivo. We want to use these little motifs. How do we really design the material to present them most effectively to govern the cell behavior? And so this, of course, has given birth to an enormous array of biomaterials designs that you'll see here at the meeting. But as we were doing this and translating things into the clinic, of course, the idea of doing this for liver remained incredibly elusive. So much so that I had an epiphany in the mid-1990s that instead of trying to replace liver in vivo, transplanted liver that was made in my lab, who would want that? What we might be better off doing with tissue engineering is figuring out how to build models of humans to understand disease and to predict how drugs would work in humans. And when Gail Naughton and I got asked to write a perspective for Science in 2002, they were expecting something about liver in a box, heart in a box for transplant, and we came up with this vision that ultimately we would maybe not even need to do these kind of transplants if we could really prevent disease or treat it early on. And this was an era where we started working on something that became known as the liver chip. It was literally a chip, a piece of silicon that was deep reactive ion etched to organize cells isolated from liver, and in this one it's rat cells, into a 3D shape so that the cells would retain a lot of the functions of liver much longer in culture than otherwise. Now this wasn't liver, but it had a lot of the functions, so much so that pharma companies were clamoring for this as a way that they might start to predict drug interactions with the liver without using animals. And over the years, we worked with about a dozen pharma companies and learned from them what they needed in their labs. So we translated this to a multi-well plate format where we use small little microfluidic pumps driven by air and vacuum in a collaboration with Dave Trumper, and ultimately commercialized that to something called the Physiomimics that's now used by most major pharma companies for predicting human responses to drugs. And there's a ton of publications, including with the FDA. But what evolved over that time that was starting out to try to predict metabolism and toxicity is an enormous interest in using human liver in these systems to model disease. And these are a couple of examples from our collaborators at Novo Nordisk. I've worked with them for about almost a decade, and they really want to move away from using animals. And you see here Lotte Knudsen, who won the Lasker Prize last year, beautiful essay describing how if they'd started with mouse genetics, they never would have gotten to GLP-1 drugs. And their VP at an NIH meeting explaining that they can't use animals because it sends them down too many blind elements. They really need human models to study cardiometabolic disease and are very strongly supportive then of our work together trying to build new models that capture more of the relevant biology. And in fact, Novo Nordisk built a whole lab right outside Boston in Watertown with about 50 people to interact with our team. And it's a team that combines systems biology and these so-called microphysiological systems so that we can start to model things like how a diabetic liver, type 2 diabetes, may attract more immune cells and what they will do in the liver. And this is an example of a project we have with them where we made a little, in a microfluidic chip using technology developed by our colleague Roger Cam initially and modified for this purpose, including aggregates of human liver cells, the different cell types, including hepatocytes and the supporting cells, in a vascular bed so that the blood vessels go through the actual aggregates and you can start to look at immune cell recruitment. And I put QR codes for the publications that are now up in bioRxiv, and many of these are on their way to being actually published in a journal if you're interested in the details. And this has, of course, sparked an enormous, there's an enormous landscape that the NIH has been supporting together with other federal agencies that they now call these novel alternative methods, meaning alternatives to animals, a combination of microfluidics and tissue engineering, but also computational analysis of patient data, phenotypic data from the clinic, together with molecular and cellular data that can be used to understand, say, genetic variations in a complex disease like diabetes. And so the NIH a few years ago commissioned two really important studies, first on how animals are being used in NIH research, and I contributed to that. Over 40% of R01s in 2020 use mice in some way or another. And then once that report was done, they decided that they really needed to catalyze alternatives to get better return on investment of the NIH money, and how should they invest in this novel alternative methods to get the community more tools that could be used. And of course, industry has been on board with this. FDA has come out saying they want to move away from animals. So there's a lot of groundswell, so much so that there's a new journal that just got started, spearheaded in part by Dan Tagley at NIH, who has supported this field of used to be called microphysiological systems and now NAMS. And so NIH is really putting a lot of effort into this, and we have been very happy to work with them along the way on what the important challenges are. Okay, so that's framing things. I want to focus on the main issue, one that derives from women's health, because that really has driven our own, what is the bar in building these kinds of technologies to use more broadly in biomedical research. And it really starts with my niece, who grew up in Atlanta. She loved school. She was a top student in her school. When she started having her periods, though, she was missing school because they were extremely painful. She would get very sick. She got put on birth control pills when she was 13. It didn't really help. Many, many tests, especially GI tests. And her female doctor eventually told my sister, you know, she's making everything up to get out of going to school. And she was 15, and, you know, lava shot out of my head, and I got her referred to a really good surgeon in Atlanta through a collaborator I had in Boston, and she had stage 3 endometriosis when she was 16. So it was a really organic disease, and she's gone on to have a couple of surgeries, and now she's fortunately has a couple of beautiful little boys. It's not that surprising that her clinician would argue with me when I suggested that Caitlin may have endometriosis, because clinicians, even to this day, were trained in textbooks like this. This is an endocrinology text that I used when I started teaching in the HST program when I was a junior faculty member. And I blew this up so you can see what it says about endometriosis patients. The typical patient will be a nulliparous patient in her 20s or early 30s. Textbooks describe her as intelligent, egocentric, over-anxious, and a perfectionist. Okay, 10% of women have this disease, it's estimated. And this is what's called diagnosis bias. So women who had the resources and the persistence to go over and over again to the doctor could get diagnosed. But I can tell you, in fact, that it's a pediatric disease, as we saw with my niece, and millions of women go underdiagnosed or undiagnosed until it's too late for them to have children. So what are we doing about it? Let me introduce you to the endometrium. It's a monolayer columnar epithelium that lines the muscle of the uterus, and so there's a monolayer columnar epithelium supported by stroma, and you can see here in a beautiful paper from Enomoto's lab the gland-like structures that emanate from the basal layer, and so the upper 2/3 is shed every month. These are clonal glands, and they accumulate somatic mutations. Anybody who wants to talk about genetics, I'm happy to do that. I'll be here till Sunday. And yet the endometrium, as beautiful as it is and life-giving, there are so many diseases that afflict women because of the nature of its constant shedding and regeneration. And endometriosis is just one of them. It is a disease that's characterized by endometrial-like tissue that grows attached in the abdominal cavity outside the uterus. Here showing on the abdomen, in the middle are 2 lesions in a patient in Brazil of our collaborator. She couldn't poop, and he took out 10 centimeters of her bowel. This disease afflicts about 1 in 10 women, and to be inclusive, we should also note girls, because it has its onset of symptoms often around the time of menarche and often starting with GI symptoms, and we still don't know the etiology. It's speculated it may be reflux menstrual tissue, but that happens in most women. Why do only some get this disease? There's also theories about it being developmental in origin. But whatever the etiology, the hypothesis is it's driven by estrogen. So therapies involving progestins to suppress estrogen or menopause drugs to suppress your whole pituitary axis are supposed to help the disease. But in fact, they don't. And patients have surgery after surgery after surgery. And these are one of my collaborators operating on a patient who's having her 3rd surgery, and we collect samples from this OR at Newton Wellesley Hospital. There's a sister disease called adenomyosis, which is when there are lesions in the muscle of the uterus. And so this particular image highlights one of the fascinating features of endometriosis for all you biomechanics people. Here's a beautifully polarized epithelial monolayer sitting in a muscle. It invades stiff tissues. And why does it do that? There's a fluid-filled lumen. You can see there. That's characteristic of lesions. And the biomechanics of this, we've only started to scratch the surface. Adenomyosis is hardly even studied. And in fact, just comparing it to Crohn's, which has a comparable morbidity, you can see from PubMed citations, there's only about 5% of the research intensity. So we have a lot of work to do. And for the young people in the room, enormous opportunity to shed light and help people live more healthy lives. So in response to this need, I got pulled in by one of the leading gynecology surgeons, shown here, Keith Isaacson, who developed a lot of the tools used in laparoscopic surgery. And he approached me and said, you know, we've been trying to develop new drugs using animal models for years and years and years. Animal models don't work. There's got to be something better we can do. So we got a foundation grant, and together with Doug Laufenberger on the systems biology side, founded a center at MIT to try to bring together all of these elements on building models of patients, studying patients, and treating patients so that we could be a nexus for how do we even start to think about engineering this disease. And so I'll tell you a little bit about what we've done. The first thing we did is realize how little we knew. There were a lot of papers describing that there's inflammation in the peritoneal cavity, in the fluid. A lot of individual pathways. RANTES is up, VEGF is up, TNF is up, in univariate analyses. We hypothesized that patients may have different molecular phenotypes. Right after we got our foundation grant, I was diagnosed with triple-negative breast cancer. And it was striking that within a few days of finding a lump myself in Singapore, I knew that my tumor was negative for 3 canonical markers related to the etiology, the prognosis, and the therapy of that particular cancer. Now, it wasn't good. I had to have supersized chemo, but it made us think that maybe there were molecular subtypes of endometriosis. So that was our first work together, was to study the whole immune networks in the peritoneal cavity of women using multiplex targeted immunotherapeutic, targeted proteomics for immune signatures, bioinformatics to infer immune pathways, and we found about a third of the patients had a signature that implicated macrophages and pointed to an intracellular signaling molecule called Jun kinase, highlighted in the immunostain images on the lower left. And Jun kinase was very excited because it was controlling inflammation and it was a non-hormonal target. Moreover, after we published this work, we discovered that there were a lot of populations cured of endometriosis by a Jun kinase inhibitor that was being developed by Merck. Steve Palmer, who was a very, very savvy reproductive endocrinologist at Merck, convinced them to do 2 preclinical models testing a Jun kinase inhibitor that was being developed for something else. Alas, Merck wouldn't do the clinical trial themselves. It's very hard to do those. They licensed it to Preglem, and the trial was not successful, probably due to potency of the drug, lack of selectivity potency, and the way the trial was designed. Now, fortunately, I linked up with Steve Palmer in Hawaii at the ASRM meeting in 2014. There was a hurricane, 12 inches of rain a day. And so we ended up together with some beer flights not shown in the picture and talked about the future of Jun kinase inhibitors. And he says, look, I'm leaving Merck, I'm going to Baylor, where they have a drug discovery institute, and I'm going to make some new inhibitors that are going to be more selective and more potent. And the thing I'm going to need to get them into the clinic are preclinical models that are human. So Linda, what you should do is you should go build the kind of model shown here in the schematic, a lesion, so the epithelia, the stroma, associated inflammatory cells, but linked to a blood vessel network so that we could look at the cycles of hormones and how they would affect immune cell infiltration. Now, we can debate who had the tougher job for when we would ultimately get back together and test his drugs in this model. So what did we do? Well, the first thing was we needed a scientific advancement, and that came in the form of Hans Cleaver's description of how to grow epithelial organoids from gut in Matrigel. That got applied to the endometrium, and so all the labs studying endometrium and endometriosis could use make tissue banks of epithelial cells by taking the glands from the patient biopsies, and this is from one of the patients at Newton Wellesley of Dr. Isaacson's. You take them down to individual cells and you grow them in these Matrigel domes with a special growth factor mix, and you can expand these and freeze them down and then reconstruct your tissue. Now, the problem though for using Matrigel for anything other than expanding these to make a tissue bank, if you're actually going to try to do the tissue engineering, love-hate relationship with Matrigel, right, for all the reasons shown here. So over a period of about 10 years, with many different postdocs and other collaborators, we developed a synthetic extracellular matrix based on some principles that Jeff Hubble introduced many decades ago, but there was an enormous amount of engineering that went into how you modify the branched pegs shown here, how you do the cross-linking, how you mix it with cells to form these hydrogels that would allow organoids to form. And what I'm going to show you is now being commercialized by Biotechne. We hope it's out by the end of the year. It took us about 10 years to develop it, and there's an enormous number of papers describing all of these individual details. But what I'll do is show you pretty pictures because the movies really show it all. From individual cells, we can now clonally grow human epithelial organoids. These happen to be intestine from Victor Hernandez-Gordia's original paper, and on the left, a matrix gel, on the right, and the synthetic gel. But you can see how they grow by pumping fluid in, creating hydrostatic pressure, and growing. Now, what is really, really valuable about a synthetic matrix is that you can tailor it to not only interact with the epithelial cells and allow them to grow, and I will say it's the first extracellular matrix that's completely synthetic that lets you grow human epithelial organoids like that. What's really, really advantageous is if you go in and start to look at what the different cell types in the tissue, not just the epithelia, but the stromal cells, the immune cells, the endothelial cells, and so on, you can tailor the components of that extracellular matrix to support multiple different cell types together so that they all can recreate their own little microenvironmental niche by accumulating matrix they produce themselves. And so shown here are co-cultures of stroma from the endometrium with the organoids, the epithelia. The green is actin because epithelia always have actin on the apical side, and the red is bimentin from the stromal cells together in this gel. And we can simulate a menstrual cycle by adding estrogen for the proliferative phase, progestin for the secretory phase when the endometrium is preparing for implantation, and then if there's no implantation, progesterone falls and you get shedding of the menstruation. And this is can be tracked by the production of a protein by the endometrium called prolactin, and when progestin falls, prolactin production goes down. So we can recapitulate these, and what is very important is stromal epithelial crosstalk is critical for how this tissue responds to hormones. In matrigel, you have so many growth factors and other things that come from that tumor matrix. It's like trying to have a conversation on the runway at Logan Airport during rush hour. You just can't talk to each other, right? You can't hear. And in this synthetic matrix, the cells can send their signals unimpeded to each other. Now, another advantage of a synthetic matrix is you can tune the biophysical properties, and as I mentioned, we're fascinated by the fact that these lesions, this one is, again, from the myometrium, and patient lesion on the left, and on the right is a light sheet image showing the epithelia invading into the muscle and a central fluid-filled lumen. And with the synthetic matrix, we can make it soft or we can make it stiffer so that those co-cultures can have the features of normal endometrium or take on invasive features like the diseased tissue. And what Steve Palmer was encouraging us to do all along was recognize that all of these lesions, of course, in vivo, are highly vascularized. This is a light sheet image that Juan Necco made when he was still a postdoc in my lab, but he's now an assistant professor in Shelley Payton's department at Tufts. And these are extremely highly vascularized. So in order to recapitulate the features of these, and a lot of them that we need in order to study the whole microenvironment for drug development, we need to adapt the beautiful approaches that many in the field, shown here is Roger Kamm's approach, for creating perfusible vessels and microfluidic devices. So in this particular implementation, and this is a commercial chip that Roger Kamm has through AIM Biotech, a solution of fibrinogen with thrombin and vascular cells, endothelial cells, and supporting fibroblasts are injected into that central channel of a microfluidic device, and there's fluid channels on either side. They form a fibrin gel, and over a period of a few days, you get these perfusible vessels. And you can see the movie slowing down now, but there are immune cells that go from one side to another under gravity flow. And so in collaboration with Roger, we got supported by NIH to ask the question, can we do this in a PEG gel? Because if we're going, first of all, if we're going to support the growth of the organoids and their functions, we can't use fibrin. We have to use a synthetic gel. And up until this point, there had never been a synthetic gel where you had perfusible vessels, certainly in a microfluidic device. Further, that gel needs to support the organoids so that we can get an endometriosis lesion model. So together with Roger, we were supported by NIH to do exactly that, replace the fibrin and figure out how do we include these organoids so that we can create a model of a lesion. And this is work by Lauren Pruitt, who gave a talk this morning and who has another talk at the meeting showing how you can adapt that gel along with fluid flow parameters to create vascularized lesions in microfluidic devices so that you can look at immune cell trafficking and so on. And it's kind of fun to watch the movie. Is the movie going to go? It's kind of fun to watch. Let me see if this movie will go. Will it go? All right, go movie. It's not going. All right, well, the movie would show you those self-assembling, those vessels self-assembling around the lesions there. Sorry, it won't show you, but Lauren can show you sometime at the meeting. She's here for the whole meeting. And now that we have these tools, Steve Palmer held up his end of the deal. He made new Jun kinase inhibitors. They are ready to go. He licensed them to a company called Cellmatics. And here we are launching a project together to look at how these Jun kinase inhibitors will act in this integrated endometriosis lesion model. And there's a lot of devil in the details things. What culture medium do we use? Does the Jun kinase inhibitor stick to the microfluidic device? All of these things have to be taken into account. And if you want to hear the details, which of course I'm not going to show you here, I encourage you to go to Lauren Pruitt's talk tomorrow because she's going to dig into those. And Jose Cadavid is also here and can speak to some of these ways that we're looking at the signaling pathways in Jun kinase and benchmarking against in vivo. Okay, so I want to kind of give you a picture of where we're going. And one of the challenges, of course, is that my lab doesn't have we have a great set of clinical collaborations, but we want to get these tools out into the community. The gel is being commercialized. We are now adapting our microfluidic device design to a format that can be driven by these air vacuum pneumatic pumps and go onto a little platform so that we can make them in a commercial setting and let our collaborators in the endometriosis field and others use them. And in fact, to try to get all of the ways that users would use them worked out at MIT before we try to build anything out in the wild, we're building a core facility at MIT for these living patient avatars and are starting to do projects with a number of labs around MIT so that we can learn all the different facets that would make the user experience better and so that we can get more information out of these. Now, I want to turn to something that I'm often called an advocate, but I really just call out the things that I see that are problems that need to be solved. And a few years ago, right after Trump was inaugurated in his first term, there were women marching around in Harvard Square with signs like this. And I stopped and I talked to them and I said, look, congratulations on your excellent menstrual health. It's really great that this is your life. But about 1 in 3 women over the age of 60 in the US has had a hysterectomy because her menstrual health was not so good. You just don't march down to the hospital and say, I just don't want my uterus anymore. You have to be really sick. And so about 1 in 5 women in the US and around the world has such problems with her period that she misses school or work at least 1 day a month, at least half her cycles during the year. And my niece is an example of that, often with diseases and conditions that need treatment, but that the patients get gaslit. And so we're on a mission to end what we call period privilege now, and to raise awareness that these menstrual problems are not something that are in your head. They're actually fascinating opportunities to learn about what governs normal and pathophysiological function of a tissue that regenerates every month in the most amazing way. Huge scientific challenges. So to that end, I got very inspired by a couple of my former postdocs, and I've been inspired by many people in the lab, but particularly Manu Platt, who, when he started his lab at Georgia Tech, focused on sickle cell disease. Now, this is a risky thing to do when you're a junior professor, but he had so many family members affected by this. He put his professional time into solving this societal problem, to the point that if I wanted to meet with him when I went to Atlanta, he told me the only time he had to meet with me was running around a 4-mile race, which I beat him that day. And he'll, I don't know that I could do it today, but I did that day. And also Shelley Payton and many others of you in the room who have championed the idea of more programming leading to this women's health SIG that's starting. And so these kinds of efforts, putting your professional time in to work on a problem and solve a problem versus just marching, I really, really love this community for what has been inspiring to me about how to get this job done. So I've started to do a little bit more education of the community abroad, and Boston Globe asked me to write a story about this a couple of years ago, and they titled it, "We Need a Moonshot," for something called menstruation science. That title comes out of a workshop that NIH had a number of years ago, and it was actually Menstruation Science and Society. NIH didn't even have a gynecology branch until 2014. And I thought it was called menstruation science. I thought, how cool is that? We can have menstruation science just like we have breast cancer science. Let's make it into a scientific language that demystifies and de-ickifies this beautiful process that has so much potential to shed light on so many other pathophysiological processes. So we had a big event at MIT, and in fact, we were able to get a gift of $10 million from Emily and Malcolm Fairbairn, alumni of MIT, and we've launched a movement that we call the Menstruation Science Movement. And it's really to start bringing together communities, first at MIT and our collaborators around the world, but also broadly here at BMES and elsewhere around solving the challenges in this field. And we've assembled a team of investigators just at MIT starting to come together of how can we bring new tools into the study of menstruation and gynecology diseases. And this just shows a picture of some of the collaborators. Dr. Mickey Tall, who moved to MIT from Stanford, has fascinating observations about the intersection between infectious disease like Lyme and chronic gynecology diseases like endometriosis. And so I just want to show you a couple of examples of work that we're doing together. We've now gotten some additional matching funding and foundation grants to build infrastructure that brings engineering tools. So it's not, studies of patients as much, although there's some of that, but to bring tools in. And one of those is the use of multiphoton label-free imaging. Shishan Yu is an assistant professor in electrical engineering at MIT. She runs the computational biophotonics lab, and some of you may be familiar with the use of multiphoton imaging to excite endogenous fluorophores in the tissue, and then you get the light out and you can look at both structure and metabolism. Here is one of her papers from a number of years ago showing imaging of a tumor and immune cell trafficking in the mammary fat pad of a mouse. So you can get the metabolism, you can see collagen fibers in green. Third harmonic shows you interfaces and lipids and immune cells. And so we have now started to apply this to study the endometrium and to study all of our in vitro models, because you don't have to do any addition of any label. It's all the endogenous light properties of the tissue. And this is our very first image of an endometrium, and her student Kunzan Liu observed that he could see the nucleoli in the epithelium. It's labeled here nucleolus. It turns out the endometrium has an unbelievably fascinating, unique way that nucleoli change throughout the menstrual cycle. They move from the center of the nucleus to the edge, and the nuclear membrane can come wrap around them, attach them to the Golgi so they export things outside the cell. This is the only tissue it's been observed in. And there's hardly anything known about it because you couldn't study it. But now we're starting to study this. And I can't show you, those are a very complicated story. We just submitted the paper and you can find it here on bioRxiv. But just to give you an example, in a co-culture, and just a droplet co-culture, if you look at the metabolic imaging of a control culture, which is maintained in media without progesterone, and then you give that duplicate sample progesterone, you see an enormous metabolic shift in the cells in response to progesterone. And I'm showing a line here to show you that the redox potential shifts very far over in response to progesterone. And there's a lot of other things going on, like the nucleoli moving around. I will tell you, though, that this is a control patient who was having surgery for some benign condition. But if you take an endometriosis patient, she doesn't exhibit this metabolic shift. Her cells don't show the nucleoli movements. And so we can now start to use these non-invasive measurements to study disease and get a lot of information content out of these cultures. I'm going to end now with a couple of more slides on a more philosophical note. So for many women, most women, the maternal instinct is a force of nature. It operates to a different degree in various people. Obviously, in me, it's a very strong force of nature. When Doug and I got married, we tried to get pregnant, and we couldn't. We failed IVF. But during IVF, it was really momentous to look at your own embryos and realize some people would make embryonic stem cells from these. I realized I couldn't do that. Moreover, I couldn't even continue working with embryonic stem cells in my lab. It just had an enormous, enormous effect on me. And the infertility is always just beneath the surface. Sometimes it comes out. But last year, when we were here at BMES and watching members of our BMES family taking such amazing leaderships in this society, particularly Cindy Reinhardt King, who was the outgoing president. She was an undergraduate at Europe in Doug Laufenberger's lab and has stayed very close to him throughout her career. Shelley Sakayama Elbert was an undergraduate researcher in my lab, and I remember at a BMES meeting, I can't remember what year, but it was at BU, her graduate supervisor got very sick. He got strep throat and lost his voice, so Shelley had to go present the poster. I had people coming up to me at the poster session asking me when Shelley was going to graduate and would she consider applying for a postdoc in their lab. They thought she was a graduate student and that she was so poised and on top of the material. And until then, she hadn't even thought really much about going to graduate school. And here she is, president of the society. Both of these women had very strong supports from their spouses, and they both raised families while managing a very demanding academic career, again, with support from their amazing spouses. And it struck us that we as a society, a professional society, but society at large, really could be more supportive of having children when you're young and supportive of young investigators in our community who can't travel to this meeting because they have childcare challenges and no money. They need their parents to come with them. They need a nanny. They need to have some way that they can afford to do both their childcare and their travel for their professional society. And this is what led Doug and me to endow a parental travel fund for young investigators. We're still working out the details of how it will work. And we hope we'll inspire others in our community who may have some resources to help these young investigators, not only in their own labs, but more broadly, travel to this meeting, engage with their peers, because they're really our future. The amazing things that I see in the talks here from students and postdocs is just completely worth it every year to see the tremendous advances. So I encourage everyone to think about ways we can continue to support our young people. I'll end there and give tremendous thanks to all my collaborators, especially my clinical collaborators, to the women who donate their cells and tissues, and to most of the members of my lab I gave thanks to. And at the end of this, I'll just encourage you all to sign up for our newsletter, because there's many ways that you, the community, can get involved to help us on this moonshot for menstruation science, and BMES being so supportive. Thank you.